Thank you, Tatiana. Good morning, good afternoon, good evening to everybody online. And it's my pleasure here to host today Yanira Mendez Gomez, Dr. Yanira Mendez Gomez. My name is Christoph Schatz. I'm a scientist here in the research laboratories of Bayer in Berlin. My work is focused on targeted radiotherapies. We're trying to make molecules which specifically deliver radionuclides to tumor cells in order to treat cancer patients. Today, I'm stepping in here in this lecture series on behalf of the Bayer Science Foundation Fellow community. And as laid out by Tatiana already, the goal of the Bayer Science Foundation is to recognize outstanding achievements in the scientific research to promote brilliant young scientists with the potential to develop their fields. In line of this goal, today we are hosting Dr. Yanira Mendez Gomez. Dr. Yanira Mendez Gomez is granted the Award of Excellence in Science in the category of Medical Sciences. I'll talk a little bit about her background. Yanira was born in Cuba. She conducted her PhD at the University of Havana in Cuba. And I understand this work was partially done in Havana, but then also at the Leibniz Institute of Plant Biochemistry in Halle, Germany. Yanira then moved on to conduct her postdoctoral work at the University of Cambridge in the UK. And I understand she's joining us today from Cambridge, where she is still pursuing her research. Dr. Yanira Mendez Gomez was granted this award for her pioneering utilization of multi-component reactions, allowing the assembly of highly complex bioconjugates, such as multivalent antibacterial vaccines or antibody-drug conjugates. And I understand today she will focus her lecture on the work she did on these conjugates and how those allow future generation of novel and more efficacious vaccines. I'm very excited to hear her lecture today, and Yanira, I would welcome you on stage and ask you to present. Welcome.
So, thank you very much, Christoph, for the kind introduction. So today I will bring you a story on multi-component reactions and how these tools allow to explore a wider and more complex chemical space within the fields of medicinal chemistry and chemical biology. But first, I would like to start with a more personal introduction. As Christoph mentioned, I come from Cuba, and I was born in a state that is located in the middle of the island that is called Sancti Spiritus. And within this state, I was born in a hometown, more properly a countryside, that I would say is located in the middle of nowhere. But being born and surrounded by such wonderful landscapes, as you see here, from the mountain to the sea, was beyond amazing. And after 18 years, I moved to Havana, where I did my chemistry undergrad, and I completed my master and PhD degrees under the supervision of Professor Daniel Garcia Rivera at the University of Havana. As you see here from most of these pictures, Cuba is what we call an eternal summer place. And then during this period, I had the opportunity to apply and was granted a DAD fellowship with which I moved then to Germany. This was a long trip from Havana to Halle an der Saale, which is a city in the eastern part of Germany, where I got to know that the year has actually more than one season. And in this place, I did and finished my PhD under the supervision of Professor Bernard Westermann at the Leibniz Institute for Plant Biochemistry. So after finishing my PhD and also part of my first postdoctoral stage, then I moved with a Marie Curie Independent Fellowship to the Department of Chemistry of the University of Cambridge, where I've been doing my research in the last 2 years under the supervision of Professor Gonzalo Bernardes. So I hope you enjoyed today my talk on multi-component reactions, which are processes that involve more than 2 molecules and to afford a product incorporating most of the atoms of the starting materials. And among these reactions, one of the most popular, I will say in the last 20 years, has been the UG4 component reaction, which involves the condensation of a carbonyl compound that is an aldehyde or ketone, and amine, a carboxylic acid, and an isonitrile to afford an N-substituted dipeptide. These reactions in general call our attention not only because of their high efficiency and diversity and complexity-oriented characters, but also because of the smooth reaction conditions in which they take place that makes them ideal tools for the modification and diversification of molecules such as proteins, carbohydrates, and glycolipids, as we will be exploring throughout this presentation. And the first opportunity I had to translate this chemistry to the chemistry of proteins and carbohydrates was precisely in the field of conjugate vaccines, more exactly antibacterial conjugate vaccines. Bacteria such as Streptococcus pneumoniae and Neisseria meningitidis, which annually account for more than 1 million deaths globally, are surrounded by an outer layer of polysaccharides, which constitute the main recognition pattern for the immune system. Nevertheless, these so-called capsular polysaccharides are poor immunogens, so they need to be conjugated to other bacterial components, such as proteins, in order to elicit a rather strong immune response. The problem arises for bacteria such as Streptococcus pneumoniae that could be surrounded by up to 90 different, and very different as you observe here, capsular polysaccharides, or what we call also serotypes. Current formulations, such as Pneumovax 23, for example, can cover up to 23 of the most invasive serotypes. But this also means 23 times the concentration of a strong immunogen, such as a protein, which often ends up in hindering the recognition of each individual capsular polysaccharide. A first solution to this problem came in 2014 by the group of Adamo, which proposed conjugating or ligating more than one capsular polysaccharide into a conjugate, in this case in which they call multivalent but unimolecular conjugates. Nevertheless, this approach, as you can imagine, is rather challenging because it involves the multiple modification in different steps of the protein, followed by the sequential incorporation of each single capsular polysaccharide. And then we came with the idea of why not using multi-component reactions. So if we come back to our slide on the UG4 component reaction, we could appreciate that most of the components involved in these reactions, that is the carbonyl compound, the amine, and the carboxylic acid, are either present or are easy to incorporate both in proteins and in carbohydrates, such as capsular polysaccharides. And more important, with this reaction, we could combine all of these components together in a single pot. In this case, our toolbox had or contained different capsular polysaccharides from Streptococcus pneumoniae, Neisseria meningitidis, and Salmonella enterica serobartifi, which we use in their original form, that is in their natural form, or in this case, for example, periodate and tempo oxidized to generate the functional groups needed for the multi-component reaction. And we mix them with either small components such as terbutyl isonitrile and other capsular polysaccharides, and in this case, hydrazine activated immunogens, in this case, BSA or tetanus and diphtheria toxoid, to either access the more simple monovalent conjugates or divalent conjugates. But to start proving our approach, we started with the most simple scenario that was, in this case, conjugating one capsular polysaccharide to hydrazide-activated toxoids. For this, we use different periodate oxidized capsular polysaccharides from Streptococcus pneumoniae, such as capsular polysaccharides 14, 7F, and 9V. And we conjugated them to tetanus and diphtheria toxoid, in this case in combination with butyl isonitrile and acetic acid. This approach allows us to generate the desired monovalent conjugates, as you see here, in only 4 hours, and with the free protein percentage and capsular polysaccharide protein ratios that were recommended by the World Health Organization. So after also proving that the recognition of each immunogen, that is the protein and the capsular polysaccharide, wasn't affected through this type of conjugation, we then immunize or administer these conjugates to BALC mice within 5 to 6 weeks of life. And then after a regime of 3 doses, we analyze the immune response of these conjugates, as you see here, in comparison with the parent capsular polysaccharides. For the parent capsular polysaccharides, as they are T-independent antigens, you see here that there is no strong immune response, as I mentioned before. But for the conjugates, it's possible to appreciate that from the second dose on, there is a strong immune response, and even for the conjugates containing capsular polysaccharides 14 and 9D, there is a strong, although non-significant, immune response from the second dose on. So with these results in hand, we then move to a more challenging task that was conjugating or having 2 capsular polysaccharides in conjugate. And in this case, we use different capsular polysaccharides also from Streptococcus pneumoniae. I'm showing here the example of tempo-oxidized capsular polysaccharide 14, but we also use capsular polysaccharide 1, which contains naturally a carboxylic acid, together with different capsular polysaccharides like 7F, 9V, 18C, 6B, etc., in which we generated a carbonyl compound through periodic oxidation. In this case, we had more or less a functional group every 7 repetitive units. And we mix these 2 types of capsular polysaccharides with terbutyl isonitrile as a small component and hydrazide-activated toxoids. In this case, also in a fair amount of time, we were able to obtain the desired divalent conjugates, in which, as you see here, they were obtained only in 1 pot, and also they were incorporated to 1 site of reaction. We demonstrate the incorporation of these polysaccharides using different characterization techniques, such as HPLC, colorimetric techniques, and also proton NMR, as you see here. But what was more important was characterizing the recognition of each capsular polysaccharide in conjugate and demonstrating that the presence of 1 polysaccharide didn't hinder the recognition of the second capsular polysaccharide, even though both polysaccharides were incorporated at a single site of reaction. Also, what was quite interesting was that the response for each particular capsular polysaccharide in this divalent conjugate was quite similar when compared to the individual monovalent conjugates that we obtained in the previous step. That is, for example, for 7F, there is a strong immune response from the second dose on, and for 14, there is a strong immune response even from the first dose on. Although this is still an ongoing research, we then moved to a different field of vaccine development, and this was the field of adjuvants. Adjuvants are substances that either increase or regulate the immune response of a vaccine, and they are of essential importance in the development of subunit vaccines, that is, formulations that lack the whole bacterial or virus composition. In this case, we are talking about, for example, protein or capsular polysaccharide or even RNA-based vaccines. And we were particularly interested in the CD1D agonist alpha-galactosyl ceramide which is interestingly an exogenous glycolipid derived from a marine sponge natural product, which is able to be recognized by the CD1D receptor on antigen-presenting cells and presented to the T-cell receptor of a subset of T cells known as invariant natural killer T cells. This interaction triggers the secretion of cytokines that are related with the T helper 1, T helper 2, and T helper 17 responses. That is, this is a rather strong and highly diverse immune response. And this is the main reason why scientists have been interested that much in modifying the scaffold of this glycolipid to either tune or bias a specific type of immune response. When analyzing the structure of this lipid or this glycolipid, we then realized that, as I highlighted here in blue, that besides those modification sites that have been previously reported, there was a chemical space that was rather unexplored, and is the chemical space beyond the secondary amide bond. In this case, we realized also that we were able to access this space when using also, once again, the UV4 component reaction, starting from a common precursor for this type of glycolipids, that is alpha-galactosyl phytosphingosine, and combining it, in this case, with a simple aldehyde such as paraformaldehyde and different carboxylic acids and isonitriles, we will be able to access this space in which we generate a kind of N-substituted peptide derivative. But also, and more importantly, we were able to access a chemical space that was rather neglected up to now, and it's the space of new alpha-galactosyl ceramide dimers, which interestingly resemble other type of adjuvants that are known as toll-like receptor 4 agonists, such as monophosphoryl lipid A. So to detail our synthesis protocols, in this case, as I mentioned before, we either started from properly protected alpha-galactosyl phytosphingosine and mixed it always with paraformaldehyde as a small component. And in the simpler approach, we employed different carboxylic acids and isonitriles to access the alpha-galactosyl ceramides, in which we included different modifications, for example, 2 extra lipid chains, and an oligoethylene glycol chain, a fluorophore, and as such, as many modifications as one could envision in this case. And also, we combine these 2 components with either bifunctional isonitriles or bifunctional carboxylic acids to access the space of alpha-galactosyl ceramide dimers. So here I summarize all of the modifications we perform. In this case, R1 is coming from the functionalization on the carboxylic acid, and R2 on the isonitrile. As you see here, we either introduce 2 lipid chains, 1 lipid chain and 1 truncated benzyl or cyclohexyl chain. We also introduce some amino acids, some oligoethylene glycol chains, which will allow us to regulate, for example, the glycolipid solubility. And in the end, we also introduced some functionalities that after final deprotection will allow us to obtain functional groups, for example, for post-modification or even for the conjugation of these glycolipids to immunogens such as proteins, as I mentioned before, or capsular polysaccharides in one of the newest approach in this type of vaccines that is called autoadjuvanting vaccines, where the adjuvant is directly linked to the immunogen. As I mentioned before, we also incorporated some fluorophores. To have an idea whether these modifications were able to affect the interaction, in this case, the ternary complex between the CD1D, as you see here in ribbon, the TCR receptor and the glycolipid, we performed several docking studies. And although you see here, there is a huge deviation from the natural or the original glycolipid, because we are introducing an extra chain. In this case, I'm showing here an extra lipid chain. The particular individual interaction of these glycolipids with either the TCR, so the T cell receptor, or the CD1D receptor wasn't affected when compared with alpha-galactosyl ceramide. Only in the case of compound 24, one sees here that there is indeed a stronger interaction with the T cell receptor. Which was interesting here was that depending on the nature of this group, whether it was more hydrophobic or more hydrophilic, then the glycolipid oriented itself in a conformation to allow an interaction with the T cell receptor, either with the T cell hydrophilic patch here in gray, or the T cell receptor hydrophobic patch. After also proving that these compounds were able to promote T cell proliferation, so antigen-specific T cell proliferation, we administered these new types of adjuvants together with the model antigen ova-albumin, and we compare the effect of these compounds with the administration of ova alone, but also with the administration of ova co-formulated with alpha-galactosyl ceramide MPEG, which is a reported and optimized analog of alpha-galactosyl ceramide containing, as I mentioned, a large PEG chain that allows, for example, intranasal administration. From the weight development and the side effects, we did not notice any appreciable change from the administration or co-administration of these adjuvants. And also from the humoral immune response, you see here that for most of these compounds, either by intramuscular or intranasal administration, they are able to elicit the secretion of IgG1 type antibodies, which are corresponding to a T helper 2 type response. In some cases, there is a rather mixed immune response, which is also characterized by the secretion of IgG2C antibodies. But which was more interesting here was that some of these compounds, I would say most of them, were able to elicit a stronger immune response or humoral immune response when compared with the administration of ova coadjuvanted with alpha-galactosyl MPEG. So in this case, to further characterize the cellular immune response, we performed different experiments. I'm showing you here only the characterization of the cytokines in the supernatants of restimulated splenocytes after the immunization. And in this case, you see that there are some compounds that elicit a strong cellular immune response. Nevertheless, this is a rather mixed T helper 1, as you see here, that is characteristic of the secretion. So we have here, for example, interferon gamma and interleukin 2, for example. And these compounds also elicit the secretion of T helper 2. So it's a rather mixed T helper 1, T helper 2 response. For some other compounds, for example, compound 3 from the intramuscular administration, this is a more polarized T helper 1 response. But as you see here, for the intranasal administration, this is a rather mixed immune response. So in conclusion, these compounds were able to trigger or elicit a strong humoral immune response and also a strong cellular immune response that was characteristic of a mixed T helper 1, T helper 2 response. So I hope up to now I was able to demonstrate that multi-component reactions, in particular the UV4 component reaction, are powerful tools for the modification and diversification of biomolecules such as glycolipids, and also for the conjugation of large biomolecules such as proteins and capsular polysaccharides even the conjugation of 3 of them in the context of vaccine development. But nowadays, the development of new multi-component processes is not satisfying the demand of these high diversity and complexity-oriented character processes. And this was what inspired the second part of this presentation and the research I've been conducting in the last 2 years, which deals with the development of new multi-component reactions. In this case, I'm presenting here today a reaction in which we combine the isonitrile tetrasyne 4+1 cycloaddition with an UV-type multi-component reaction, such as the UV4 component reaction, as mentioned before, to access a novel chemical space of pyrazole-amide derivatives. Isonitriles and tetrasynes undergo a fast inverse electron demand 4+1 cycloaddition followed by a 4+2 cycloreversion to a 4-pyrazole imines derivatives such as intermediate I2 here. These compounds, in this case I2, are stable only if one starts from a tertiary isonitrile, as represented in this case, which has been exploited on the diversification and modification of, for example, isonitrile-derived proteins with a fluorescent probe, which was reported by the group of Lipper. But there is a problem when the isonitrile is indeed, or instead, a primary or secondary isonitrile, because then intermediate I2 rapidly tautomerizes to what we call an aromatic imine, in this case, intermediate I3, which in turn experiments a rapid hydrolysis, even in the presence of water traces, to the corresponding carbonyl compound and a pyrazole amine. This, of course, have been also employed or exploited on the masking of several functional groups, such as aldehyde and even native proteins. But this is the end of story, to be honest, when mixing a primary isonitrile and a tetrasyne. But then we kind of look closer to this type of reaction, and also based on our knowledge on the UV4 component reaction and the main mechanistic step that involved the addition of a carboxylate ion and an iminium ion, in this case simultaneously, to an isonitrile. And then we realized that we already had the isonitrile from the reaction with the tetrasyne, and the iminium ion, in this case, could be originated from the interaction of either one of these intermediates, I2 or I3, that is, there are 2 imines, and the reaction with the carboxylic acid. So in this case, we could mix a second equivalent of the same isonitrile or a different isonitrile, a carboxylic acid, and the product deriving from the reaction of the isonitrile and the tetrasyne. And in this case, access a space of compounds that are similar to those obtained in a UV-type multi-component reaction, which are indeed stable compounds. For proving this concept, we started mixing dipyridyl tetrasyne, in this case, with N-butyl isonitrile in a first step as a model for a primary isonitrile. We left this reaction to occur for 90 minutes, and then we added a second equivalent of the same isonitrile and acetic acid. And we weren't sure at this moment which one of these intermediates will be reactive. What we were sure of is that there was a color change from the in this case, bright fuchsia that is characteristic of the tetrasyne to a light yellow that is characteristic of the occurrence of this first reaction. And then later to a bright orange after we added the carboxylic acid and the second equivalent of the isonitrile, followed by the appearance of a kind of light yellow precipitate. And after 40 minutes, we saw the complete conversion to a product having the molecular weight corresponding to one of these 2 products, or probably to the 2 of them. But we weren't sure, as I mentioned before, so we had our hypothesis that probably mixing all components together from the first step, like in a kind of in situ scenario, we were able to catch the reactivity of intermediate I2, whereas adding the components separately, we were able to catch the reactivity of intermediate I3. Nevertheless, the two products isolated from these two scenarios had a unique structure, and this structure matched only to the structure of the compound derived from intermediate I3. To better understand this bias, we then perform NMR kinetic studies on the first step, that is the tautomerization step, or the first reaction, using dipyridyl tetrasyne and butyl isonitrile. And as you see here, from the different signals identified, these signals, even from the first minute, only corresponded to intermediate I3, whereas no signal corresponding from to I2 or to I1 were observed during this experiment, meaning that this reaction is so fast, even the tautomerization step, to be, in this case, monitored in the time scale of the NMR experiment. Also, after 20 minutes, the amount of intermediate I3 almost equals the amount of the starting material, in this case, the tetrasyne, which is a good indication that we could add, for example, the carboxylic acid and the isonitrile of the multi-component reaction at any step of this process, that is in situ, after 10 minutes, 20 minutes, or even after 90 minutes, as we performed before. To analyze or to extend the scope of this reaction to other tetrasynes, we use, for example, along with two pyridyl tetrasyne, 4-pyridyl tetrasyne, or phenyl substituted tetrasynes, methyl, 2-pyridyl, and dimethyl. And in this case, what we observed was that those tetrasynes substituted with the 2-pyridyl group allowed to obtain the product in a time frame from around 2 hours to 4 hours approximately, whereas those tetrasynes, lacking this 2-pyridyl group, were not able to afford the product until more than 68 hours. The only compound that didn't afford any product was the 4-pyridyl substituted tetrasyne, which has been described as a very unstable compound. In this case, we wanted to understand what was the difference and why these compounds were so less reactive. And we also performed NMR kinetic studies in which we mixed diphenyl tetrasyne and butyl isonitrile. And what we observed was that even after 37 minutes, there was possible only to observe intermediate I2, whereas intermediate I3 started to be formed only after 4 hours. Even a significant amount of this intermediate, as we remember for the case before, is the reactive one, was only possible to be observed after even 14 hours. So this kind of explains the kind of poorer kinetics of this type of tetrasyne in comparison with the pyridyl substituted one. To better have a hint on this difference, we then performed density functional theory studies on the tautomerization step. And indeed, we were able to demonstrate that the nitrogen atom on the pyridyl ring assists the CH deprotonation and the formation of a pyridinium sweeter ion, in this case, which will lead to a final intermediate I3 through a hydrogen transfer. Similar reaction route is not possible for the diphenyl derivative, in which it takes a different route to go through an iminium sweeter ion in this case, but the transition state has a rather high energy. So this route is rather not possible. Only incorporating some water molecules to assist this hydrogen transfer did somehow explain why this reaction was taking care after some time. After demonstrating the scope of this reaction for different tetrazymes, we then moved to use different isonitriles, and we proved this reaction with secondary or tertiary isonitriles. From the use of cyclohexyl, that is a secondary isonitrile, we observed that the secondary imine form was completely unreacted, as already reported for some secondary imines in the UGI type multi-component reactions. And in the case of the tertiary isonitrile, as I mentioned before, this formed a rather stable intermediate I2 type product. That means this intermediate is locked and there is no possibility of the formation of I3. Of course, in this case, there was also it was also not possible to have a reaction. Nevertheless, mixing or having a primary isonitrile in the first step, followed by the addition of a secondary and a tertiary isonitrile, did allow us to obtain the desired pyrazole amide derivatives. In this case, also extending the reaction scope to more hydrophilic isonitriles or tetrazymes, and even larger hydrophilic or hydrophobic carboxylic acids was possible, and we were able to explore the possibility of employing different solvent systems, or here more unpolar or more polar here, where we utilize, for example, trifluoroethanol or even buffer systems. And kind of to demonstrate the scope of this reaction in more complex systems, we then moved to the modification of randomly generated and on solid phase a synthesized peptide, in which we place a cysteine and then after introduce through Michael addition, for example, a tetrazyme or an isonitrile as a starting material. And then we mix it with, for example, in the first case, with a carboxylic acid and an isonitrile, and in the second case, with the tetrazyme, the carboxylic acid, and the isonitrile. And this reaction proved indeed efficient for the solid phase modification of peptides, which would later extend to the modification of a p53 peptide activator in this case, where we introduced a tetrazyme reactant group in different positions of the peptide, not only to prove that this modification was possible in the context of only introducing a pyrazole amide, but also in the context of stapling this peptide, as we demonstrate here, and stabilizing an alpha helical conformation. Also, for example, using a more meaningful modifications, such as the introduction of fluorescent carboxylic acid, did allow us to study the internalization of these peptides using HCT 116 cells and after 4 hours treatment, which was quite interesting. So I hope I was able to demonstrate that this multi-component approach allows us to convert a rather simple two-component reaction that didn't afford a stable product into a multi-component process that allows us to obtain a novel chemical space of pyrazole amide derivatives, which is of great importance in the field of the development of new multi-component reactions. So with this, I would like to thank all my mentors and collaborators throughout these years. So from the synthesis lab in Havana, Professor Dr. Daniel Garcia Rivera, which was my first supervisor, and Dr. Ana Rodriguez Unpierre, which worked along with all the time in the development of vaccine conjugates. From IPV Halle, Professor Dr. Bernard Westerman, which was my second PhD supervisor, and Professor Dr. Louis Gervais Johan, Dr. Melida Bari, and Mohamed Youssefi. From the Helmholtz Center for Infections Research, Dr. Thomas Evensen, Kai Schulze, and Professor Dr. Carlos Guzman from the University of Cambridge, Professor Gonzalo Bernardes, and Dr. Aldrin Vasco Vidal, which has been one of my main collaborators throughout this year. And from the Finland Institute of Vaccines, where most of this study started, the story of conjugate vaccine development. Professor Dr. Vicente Pérez Bencomo, Dr. Dagmar Garcia Rivera, Dr. Hanoi Calderon, and Abel Sanui. Also to Dr. Gonzalo Jimenez Oces and Tiago Rodriguez. And I would like to thank you for your attention, and I'm more than happy to answer any questions that you have.
Thank you so much, Yanira. Tatiana, you have to guide me a little bit. So do we do the Q&A in this round, or do we move on to another? Well,
yeah, we do the Q&A now, so in the official part, and then there's also a more unofficial part later on where people can maybe also have more detailed questions around Yanira's development, career journey, and so forth. So, but here is now the science questions if you want. So everybody who has a science question can now ask the questions in the chat.
Okay, okay, then I also open my chat so I get the chance to read when there's something there, or you just speak up. But in the meantime, maybe I can start with a very personal question, if you don't mind, Yanira. I do have an appointment with my doctor tomorrow to get a flu shot for this year. So I understand that your multi-component reaction allows for a more diverse, more immunogenic and maybe more efficient way of doing vaccination. I'm curious, is there a chance that next year I will not rely on a traditional flu vaccine coming from fertilized chicken egg, but from your multi-component reaction approach?
Oh. So no, I think we're a bit far away from, let's say, applying these multi-component processes. So although I start developing this process already in the industry, so I think this is rather challenging. So I hope in the future, industry will be interested in actually using this type of processes because it's quite efficient and allows to obtain a vaccine conjugate just in few hours. And also we were able to upscale this reaction and prove that was efficient for even larger scales. But I don't think that probably next year. So hopefully in the future, people will be able to appreciate the importance and also the possibilities of this reaction in generating high complexity and also facilitating this type of conjugation.
Okay, and understood. And sure, next year is maybe a little bit too challenging. But if I may follow up on the potential advantage, so I understand, or maybe you help me whether I understand it correctly. So the advantage is twofold. On the one hand, to this chemical process, you would have the potential to be faster than the traditional process going through fertilized chicken egg. Is that true?
Well, I mean, yeah, they have the potential to, in this case, compared to other chemical procedures, I think it's not that it's faster, but it allows to combine these multiple components, in this case, these multiple polysaccharides in a single step, which is, I will say, faster in this case, because otherwise other approaches involve, for example, obtaining the protein, modifying the protein, and introducing the different functional groups, and then step by step, which means each step implies, for example, an extra step of purification or cleaning, and then adding where one also lacks a big part of the component. So I think it's more efficient in this sense. So I would say so.
What about another angle, which is, usually for this vaccination, they're trying to improve the variability, do 2 different strains, 3 different strains, what they do for the flu right now. Would that be also something where your approach could come up with a 4-fold or 5 strains or whatever, so to have that basically cover
Yeah.
more ground with this approach?
Yes, and actually that was the main idea. So formulations or multivalent formulations, as I explained, for example, for pneumococcus or Neisseria meningitidis, they cover the different monovalent, that is each capsula polysaccharide conjugated to a single protein. But of course, it's not possible to cover all of them in a single formulation because while adding strong immunogens such as a protein each time in each one of the conjugates, this diminishes the recognition of each particular and individual capsula polysaccharide, for example, or other type of immunogens. Whereas this reaction will allow to combine different molecules all together and have indeed, let's say, a conjugate that already contains the characteristics of each. For example, let's say in this case, I show only divalent, but it could be trivalent, or they have like more than 3 valencies in even one molecule, which will allow also, like as you mentioned, not only to let's say have like a broader coverage, but also to exchange the coverage really fast. Because one of the problems of, for example, bacteria such as Streptococcus pneumoniae is that varies from region to region, and also the most invasive serotypes are constantly changing. That is, each formulation needs to be kind of updated every time.
Okay, I'm just looking at the chat and I see one question from someone, DA. I'm reading it to you, and then please, whoever put this down here could then also speak up. So the question is around the solid phase multi-component reaction, and the question is, did you need MW assistance? Which mixture of solvents did you use?
Kind of didn't get the question.
Well, also I have to admit that I don't know what MW stands for, but I guess it was wondering about the specific chemical reaction you did use for the solid phase reaction.
Microwave assistance.
That
yeah.
might be it.
Yeah, I'm reading it now. No, not for the solid phase modification of peptides. I did not need microwave assistance. This reaction were carried out overnight. So, well, based on the previous experience our group or our collaborator had in the on solid phase development of multi-component reaction. Sorry. And we did perform them overnight. We use a mixture of dichloromethane, that was the main solvent, but we also added some drops of trifluoroethanol to help solubilizing the components. And in this case, we discovered for some of these reactions, not all of them, that having later, like a mixture 1-to-1 dichloromethane trifluoroethanol was helping the reaction to occur faster. So in some cases, even after 2 hours on solid phase.
Thank you. So other people, please speak up if there's more question. Otherwise, I follow up with another one, if that's okay, Yanira.
Yes.
So for your story where you discussed the a vaccination as an intramuscular versus intranasal approach, which obviously looked very promising, and I understood that it appeared that this multiple component reaction derived vaccine may allow intranasal application, whereas I guess traditionally the intramuscular is done. And my question is, do you expect this to be a general phenomenon, so that in the future, if everything goes with MCR, we will only use intranasal? So if I go to my flu shot in 10 years down the road, I will have it in the nose and not in the arm. Is that what I can expect?
Well, I mean, this is, I won't generalize for multi-component reactions or the process we employ for the generation of these adjuvants, but I think in general, independently from the chemical process employed for the generation of the adjuvants, there is a tendency to generate more soluble adjuvants, as I mentioned, the previously reported galactosylceramide and PEG, to aid this type of intranasal formulations, because, yeah, as we know, for example, from COVID, most of pathogens, so the way of entrance to the organism is mucosal, actually. So this type of immunization could be of great help in the future. So there is a high interest right now in the development of intranasal formulations, and having these results particularly, but just because we were able to diversify these molecules in a way that it allows us to either use one type of administration or the others, I think is quite promising.
Thank you. I don't see anything. Tatiana, you please notify myself if there's something I'm missing. Otherwise, I'll ask my final question. You know, I'm working in the field of cancer therapy. Here we also, there's a long history of people trying to generate vaccines to treat cancer, so far with little success. Do you think MCR may make all the difference, or have you considered applying this concept to the field of cancer?
I mean, why not? So I think part of our story that I didn't present today on antibody-drug conjugates, for example, a big part of this story also employs multi-component reaction for the generation either of the, for example, the drugs employed in the conjugates, or also for the conjugation processes. Also, I think MCRs allow to synthesize, as I mentioned, like more diverse and complex linker systems that could be used for the different applications, and in this case also for the development of anti-cancer vaccines. Why not? I think these processes in general have a big advantage in this field.
Thank you. I read here in the chat that there's a question from Anna. Anna, please ask your question.
We cannot unmute the people, unfortunately.
Okay, so how do we find this question?
So the biggest synthetic difficulty. So the question about is about she's wondering about where did you encounter the biggest synthetic difficulties? Is there anything you can share here into this? So what was the most challenging part in the synthetic? Or are there any, maybe? I don't know.
Yeah, so there's always many synthetic challenges throughout these stories. For example, in the case of the glycolipids, I would say this was rather challenging at the very beginning, because these are substances that are rather unpolar. And in the end, one can obtain substances that are a bit kind of amphipathic, so they form detergents and so on. So yeah, kind of optimizing the reaction conditions for an efficient obtention of these derivatives was rather challenging, I would say. Yeah, I don't know. Also, when we try to develop the multivalent conjugates, so optimizing the ratio of polysaccharide to protein to be employed in this type of reaction was rather challenging. So I did not mention it here, and I don't know if I can go back to the slides. Let me see. Well, so it seems here that we have like site-specific functionalization of a protein. So, and that's it. But this type of molecules, they actually, as they contain a functional group, like every 7 repetitive units, for example, they will allow for multiple reactions. So in the end, one has like a mesh. So where we have like, for example, a protein that is linked to the 2 polysaccharides, but this polysaccharides already in the 7th repetitive unit or probably in the 14th already have another reaction site. So having this type of mesh as products in the very end is rather challenging because one needs to really optimize the ratio of polysaccharides and proteins in the final reaction setup to obtain actually the required ratio that is the one that is recommended from the World Health Organization, that is from 2 to 1, that is capsular polysaccharide protein, to 1 to 2 protein capsular polysaccharide in this case. So that was also rather challenging.
Okay, I'd say thank you very much. Tatjana, if you agree, then again, thank you very much, Yanira, for this excellent talk. We conclude the Q&A session here, and Tatjana, maybe you guide people to the rest of the session, where to go and what to do.
Yeah, thank you so much. Yeah, thank you also from my side, Yanira, for this interesting talk. Yeah, so many different reactions and so forth, and yeah, very interesting also to see how this can influence immune responses and enhance them. So very